Today I want to wrap up the 2.4 notes on macromolecules by reviewing some of the important ideas that you need to know for your upcoming Unit 2B test. I want to start off by reviewing some of the prefixes and suffixes that are very common in science terminology. If you come across a word that you don't understand, it's often useful to remember these root words and can help you get some meaning out of the term even if you don't remember the exact definition. So what you can do is pause this video and quiz yourself on these terms and then check your answers as we go along. First we have micro, which of course means small. So your example here is your microscope, which allows you to see small things. The opposite of micro is macro, which means large. And of course the main idea of this unit are the macromolecules. These are large molecules made out of many smaller molecules. Next, we have a series of numerical terms. And remember, in science, we sometimes use Latin and sometimes use Greek. And that's why there's two of some of these, because some of these root words are Latin, some of these root words are Greek, but they mean the same thing. So mono, or uni, means one. So a monosaccharide, remember, is one sugar. Di, or bi, means two. So of course we have a disaccharide, which is two simple sugars bonded together. Then we have tri, which of course means three. Think about a triglyceride, has 3 fatty acids. Then a term that we didn't specifically go over was quad or tetra, which is the next number in the list, which means 4. A quadruped is an animal that walks on 4 legs. Then we have penta. So we use that in the example pentose, which is a 5-carbon sugar. Then hexa, which means 6, and we have a hexose, which is a 6-carbon sugar. Then we have the term poly, which means many. So if we think about a polypeptide or a polygon, okay, so this is where we have many of something. Then we have some common suffixes in biology. So remember, ose is representative of a sugar or just a carb in general. So we have the example of glucose or sucrose. Then ase is the ending that we use to denote an enzyme. Lactase is the enzyme that breaks down lactose. Or amylase is the enzyme that breaks down the starch amylose. Then one that we saw but didn't specifically point out is the ending in is actually used to denote a protein. So a lot of protein names end in in. So insulin or hemoglobin are both proteins that your body produces. Then finally we have ol. This is representative of an alcohol. So the example we saw was glycerol, which is the backbone of a triglyceride. Next, we want to review the macromolecules themselves. Remember, there are 4 macromolecules we need to know: carbohydrates, lipids, proteins, and nucleic acids. For each of these macromolecules, you need to know 3 things. You need to know the monomer, that is the building block, the polymer, that is the structure that you're making with these building blocks, and then its function, the use inside of the cell. So again, you can pause the video and quiz yourself on each one of these to make sure that you know all 3 of these items for each of these macromolecules. So let's start with the carbohydrates. The building blocks of carbs are the monosaccharides. And some good examples of those would be glucose, fructose, and galactose. These are simple sugars, or a single sugar molecule. Mono, again, means one. Then our polymer of the carbs are the disaccharides or the polysaccharides. Again, remember the term saccharide is in reference to a sugar. So disaccharide is where we have 2 sugars, like in sucrose or lactose. And a polysaccharide is where we have many sugars, like starch or cellulose. And the use of the carbs in the body is mostly as a source of cellular energy. So this is the fuel for your cells. But also there are some structural components such as cellulose, which is the building block of plant cell walls. Next, let's look at the lipids. The monomers, the building blocks of lipids, are glycerols and fatty acids. When you put those together, you get the polymers, namely the triglyceride. But also, if you throw in a phosphate group, you could get a phospholipid. Inside the cell, lipids have many different uses, one being for storing energy, because lipids don't break down very easily, so you can store energy for a long period of time in the form of fat tissue. Then you have protection, insulation, and even chemical messages in the form of steroids. Next, let's look at the proteins. The building blocks of proteins are amino acids. Remember, there are 20 common amino acids that we come across in building proteins. Again, it's not necessary to memorize all the different amino acids, just know that amino acids in general are the building blocks of proteins. When you chain several amino acids together, you get what we call a polypeptide. Remember, the peptide bond is what holds the amino acids together. So if you have many amino acids, you have many peptide bonds, so we call that a polypeptide. Now remember, a full protein has many amino acids and then is going to have a complex structure that is caused by the chain of amino acids being folded and shaped in a very particular way. Remember, proteins have a lot of different uses, and we categorize them in two different ways. We have the structural proteins, which are also known as fibrous proteins due to their long thread-like structure. These are the proteins that are used as building blocks to actually build the structure of your cells. Then we have the functional proteins, which are sometimes called globular proteins due to their more or less spherical shape. And these are the proteins that have a specific job in the body. And the two that we talked about in particular are the hormones, which are chemical messaging proteins, and the enzymes, which help regulate chemical reactions. Finally, we have our nucleic acids. The building blocks of nucleic acids, of course, are the nucleotides. And you need to know that the nucleotide has three components. You have your phosphate group, your 5-carbon sugar, or a pentose sugar, and then you have your nitrogenous base. Remember, it's the nitrogenous base that differs from one nucleotide to the next. The polymer of a nucleotide is a long-chain nucleic acid, and the two main examples we have, of course, are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. And of course, these nucleic acid molecules play a very important role in storing information in the cell. Hopefully this overview is helpful. Make sure that you go back and review your notes so that you can remind yourself of all the details of all these different molecules that we need to know.